I'm truly excited to dive into the latest research today. Every year, we have countless new technologies and tools that have the potential to change health and humanity for the better. Many of them seem straight out of fantastical TV shows and movies, giving us hope and fueling our dreams of the future. Today, we're going to talk about a topic science fiction fans may be familiar with: gene editing. Thanks to technological advances, gene editing has become a powerful, real-world tool for studying and potentially treating disease. Today, we'll be joined by VAI's Dr. Hong Lee, an expert in CRISPR-Cas gene editing technology, for a crash course on gene editing and how it is changing the game in human research. I've had the pleasure of working with and supporting Dr. Lee since she joined VAI in the summer of 2024, and I'm delighted for you to hear more about her work today. She leverages CRISPR, cryo-electron microscopy, and other leading-edge technologies to explore the mechanisms underlying RNA-mediated processes. She has built a multifaceted research program that has informed new strategies for gene editing, cancer diagnosis, and virus detection. We'll have time for questions after Dr. Lee's presentation, and we encourage you to post your questions in the chat. In the meantime, please join me in welcoming Dr. Lee.
Thank you, Mary, for that introduction. I welcome everyone to the public lecture on gene editing. So this talk will be divided into 4 parts. If I can advance the next slide. Then I'll provide an introduction to the talk and leading to the CRISPR technology, and I will describe what is the CRISPR technology, and then I will provide some examples of what we study here at VAI, and then I finally provide topics and for discussion. And so the first, I wanted to introduce myself a little bit. Next slide. That I moved to here, as Mary said, last year to VAI. Most of our studies is discovering as well as characterizing and repurposing CRISPR technology as well as related technology for both fundamental biology studies as well as a potential therapeutic use. So prior to joining VAI, I was a professor of chemistry and biochemistry at Florida State University. When I was, and as a graduate student, actually in University of Rochester, studying at the interface of physics and biology, that inspired me to the research that I'm carrying out today. While reading the book of The Double Helix by James Watson, and James Watson and Francis Crick used a technology called X-ray diffraction, shown here as taken by scientist Rosalind Franklin, of what we call a diffraction of this mysterious DNA molecule. That led them to the elucidation of the 3-dimensional shape of this DNA molecule. That really started this field of molecular biology and started an exciting journey of understanding the DNA and led the foundation to today's genome editing. And I was fortunate enough at Florida State and become the colleague of Professor Don Casper, who was a dear friend of Rosalind Franklin back in Cambridge, England. And my postdoctoral training was in Caltech, where under the guidance of Professor John Abelson, who introduced to me this equally exciting molecule called RNA. That is also a foundation to the discovery and use of CRISPR technology. So if the mid-20th century was a DNA revolution and the discovery of CRISPR technology would be one of the RNA revolution. So next slide. Many of you might know that the DNA is actually the molecule of inheritance, and that it gave rise to the living organism instructions as well as inheritance. But many of you may not know that the discovery was made by studying this simple system, phage, how it infects the bacteria, and by mostly three scientists, Max Debroik, Alpha Hirsch, and Martha Chase. And then they use this simple manipulable viruses, phage, to really firm the discovery that the DNA is the hereditary molecule. And phages are the viruses that infect the bacteria, much like the flu viruses that infect the humans. And so, and this discovery set the stage of the decades of research on what the DNA, how does it give instruction to the cell, and how does it, when it's going wrong, cause diseases. So the next slides, please. So one of the first study was to what is actually DNA molecules made of, and then they are made of what we call these building blocks, nucleotides, that's sticking together by by this what's called a phosphosugar backbone. And these nucleotides, it has complementarity to the opposite strand of the DNA, much like the zippers when they're trying to be zipping together. So they always come in pairs. And so next slide, please. And in simple organisms such as bacteria, the DNA molecule, the genome, or we call the genome, that comprise the DNA molecule are pretty simple. They're usually circular, double-stranded circular DNA molecules, free of all these higher order structures. But in animal and plants, the DNA molecules are double-stranded, but wrapping around protein molecules that they call the histones, and to form these higher ordered structures. So in addition, the genome has a lot of diversity. And for instance, in higher eukaryotes, such as animal and plants, they are actually have additional chemical modifications on the DNA, as well as histones, and we call these epigenetic modifications. In addition to the sequences of these nucleotides and give instruction to the cell, these epigenetic modifications, as well as DNA modification, that further alters the signal of the genome, and that can cause different biology, as well as, when it goes wrong, cause diseases. So to transfer the information, genetic information, that the DNA usually have to make another molecule according to the nucleotide sequences called RNA. Some of these RNA molecules actually give the instructions to the cell to make the molecules called the proteins. And this principle is called a central dogma. That's true for all living organisms, that the protein molecules, obviously what we call the workers of the cell, they make up our tissues, they make up skins, bones, and just the structure, as well as acting as enzyme, for instance, for digesting food. So therefore, the instruction given by the DNA can direct what is the property of these proteins. And so therefore, if there is a change in the DNA sequences, you could change the RNA and the protein, which give rise to different traits of the organism. For instance, a different color of a plant. So much like one changes the letters in a sentence, that will give rise to a different meaning. And so, given the importance of what we call the information that's encoded in the DNA, and scientists always wanted to say that we wish there is a way we could rewrite the code of life, meaning we can change the code of the DNA, that will enable us to, first of all, to study the fundamental biology. If you can alter the DNA sequences, that's the blueprint of the organism. And you could also use change the DNA to correct what we call the mutations in the patient genome. And you could also, in fact, to remove certain genes, certain stretch of sequences, or even add certain stretch of sequences that can change the traits of the organism. You can also maybe influence in genetic information without even changing the sequence, and such as these epigenetic modifications of the genome. So despite that, this has been desired for many years prior to the discovery of this new technology called CRISPR, there was just no good, safe, and convenient, simple methods for doing so. So we lacked such technology. So excitingly, it is in around 2012, this enzyme called a CRISPR technology was born. I'm showing this phage and bacteria system again. And so the study of the phage and the bacteria, the virus infecting the bacteria, led to the discovery of the CRISPR. So in fact, the sort of first landmark study was made by studying the yogurt, the bacteria that made in our yogurt, culture our yogurt, that the company who studied this bacteria wanted to make sure the yogurt is not infected by the viruses. And then they discovered that there existing a mechanism in these bacteria that they can fight the infecting phages. And this mechanism is called a CRISPR. And then the CRISPR is a stretch of DNA sequences in many, many bacteria that they encode a series of CRISPR enzymes. So these are the enzymes that could, upon the phage infection to the bacteria, they can restrict, they can kill these viruses so that they can survive. Much like the human antibodies that we develop that allow us to fight the viruses, flu viruses or CoV-2 viruses. So it is really the CRISPR enzymes is native to bacteria. They really are used, these enzymes, to fight the infection of their own infection. And this is the enzyme that the scientists studied to the detail and then discovered that this enzyme could be taken out of the bacteria and use it for genome editing. And so why is this enzyme are able to do that? And so there are many CRISPR enzymes, among which there are two that are mostly used in genome editing. And so we gave them names of a Cas, CRISPR-associated proteins, Cas9, Cas1, Cas2, all the way to, Cas14 now. So these two enzymes, Cas9 and Cas12, are enzymes the bacteria uses to cut virus DNA. And then so that comes back to why we can use the CRISPR to alter the DNA sequences in genome. Especially the Cas9, which is the most widely used genome editing enzyme. So these blocks really shows the shape in a really cartoon representation of how the enzymes are made of. So they made of several parts. One of the parts are the ones that's holding the DNA, as you will see later. But these green and blue parts are representing what we called the active sites of the enzyme. So these are the sites where they actually can break DNA phosphasugar backbone. So as I introduced earlier, this molecule called RNA molecule, that many of these RNA molecules that's copied, made by the DNA, they do not encode the proteins, and these are called non-coding RNAs. So CRISPR enzymes actually widely use these non-coding RNAs, that they stopped being as the RNA as a functional molecule themselves. So they work with the protein molecules. In this case, they could work with Cas9 molecules. So they would partner with the Cas9. So obviously the significance of the discovery of this Cas9 was the 2020 Nobel Prize in chemistry by given to Emmanuel Charpentier and Jennifer Doudna for its potential in many research as well as therapeutics. So RNA molecules that's made also by the CRISPR locus in bacteria, it has this remarkable property. First is part of this RNA that's going to partner with these Cas9, Cas12 protein enzymes came actually from virus themselves. So they remember the information of the unique code that in the phage molecule that previously infected them. By bearing this information of this DNA, of the sequences that in the phage, it could allow this RNA to recognize the phage in a subsequent infection. So that's the first remarkable property. And so in phage, if the Cas9 molecule were to cut the DNA from the phage, it would unzip the phage DNA, which are double-stranded, but instead replace one of the DNA strands of the phage by the RNA that it remembered and that now become part of the Cas9 enzyme. And that allows this Cas9 to be properly sitting on the phage DNA and break the chemical bond of the phage DNA, allow it to render its non-functional. And what's remarkable about this RNA, we also call them a guide RNA because they sort of take the Cas9 enzyme to where the phage DNA is. And so it knows not to cut its own DNA. That will be devastating. And then so we call them a guide RNA. What's remarkable of the guide RNA of the Cas9 as well as Cas12 is you can change it when you want to use for a different purpose. That's made this Cas9 technology what we call a programmable, meaning if you are synthetically making this guide RNA for Cas9 enzyme, but dial the sequence of the guide region of the guide RNA to a sequence, let's say match part of the gene of in the human genome, and you would be able to take the Cas9 or Cas12 enzyme to the human gene in the genome. And that's what makes this technology powerful. And it's such a simple technology that allow one to realize. All right, And so obviously the realizing that this is the potential for the Cas9 and Cas12 to be used in biotechnology, and that set the stage for the widespread application of this enzyme. All right, so there are different ways one can use this RNA-guided double-stranded DNA break enzyme, Cas9, as well as Cas12. So the very first mode of the use of the CRISPR technology would be to direct the Cas9 enzyme to a part of the genome using a guide RNA that you dial it to, and you can make a double-stranded break. So the double-stranded break, we call it cutting, that triggers in the human cell and called DNA repair. So the human cell has the machineries that the minute your DNA is broken, it'll try to stitch them together. And so that's called the double-stranded break repair. So the way the cell repair our genome, there are two ways to repair it. One way is just to stitch them together as best as it can. But the way when it is stitched together usually will change, they alter the original sequence of the DNA. Therefore, eliminating the original message that's encoded in the DNA. So in that case, one would be able to destroy that message. So there are also another way of repair in the human genome, which is to supply a new DNA, and that we called a template DNA. In conjunction with double-stranded break, with a new DNA template, one can actually repair it according to what the new sequence encoded in the template will be. And that will allow us to alter, for instance, the disease mutations and unwanted changes that's happening in the genome. So this is the very first rudimentary direct application of the CRISPR-Cas9. However, because of the programmability of the CRISPR-Cas9, many CRISPR-derived technology has been discovered. These are the technology that does not involve in double-stranded break. Some of them just breaks one, we call it a nick on the DNA. So these CRISPR-Cas9 mediated technology, they are usually a fusion of the enzyme. So it's like taking a CRISPR-Cas9, attaching to another tool to the Cas9, so that you can apply additional changes in the DNA. For instance, there are technology called base editors. So that means not to make a cut in the DNA at the location that guide RNA led you to, but rather to change the nucleotide to another nucleotide, to swap the nucleotide sequence. This is also powerful, but much safer way to do that. One can also use it to attach to other enzymes to make the CRISPR, such as what we call a prime editing, and that can actually insert a stretch of a DNA, although short piece of a DNA at the site when targeted to. You can also, not cutting anything on a DNA, but take the CRISPR-Cas9 to the genome of the location that you can turn what we call silencing certain genes, or you can activate certain genes. In other words, interfere with the RNA production or the subsequent protein production. So realize the goal of programmed changing of the downstream product of the gene product. And one technology has also made it possible, for instance, to guide the CRISPR-Cas9 to the genome of the interest, to add these chemical modifications or to erase these chemical modifications which in turn can turn gene on and off. So many CRISPR derived technology that are equally exciting and advancing their applications. Interestingly, so far, all the CRISPR-Cas9s or Cas12 they are not able to sense the chemical modifications on the genome. And so despite the fact we know the fact, which is one of the focus, the study area of Van Andel Institute, that the DNA methylation are incredibly important in fundamental biology as well as in the development of diseases. But none of these CRISPR technology that is not able to sense the chemical modifications on the DNA. So currently lacking these set of technology. One thing scientists are very cautious about is one mode of the CRISPR function. That's called off-target cutting, which means that there are homologous regions of the genome that's to the region you programmed it to. And there is a very, very, very small chance, but not zero, and that the CRISPR-Cas12, 9 and 12, would be able to make break. And this is what we called off-target cutting, and obviously it's undesired, and it is some activity one the scientists are paying a lot of close attention to, and to try to eliminate them and to make the technology more precise. And so one of our research goals, for instance, is to achieve that by utilizing the methylation markers on the DNA. Because of this technology, I believe it's still young. It's about 13 years ago, and that's started this discovery. And it's moving very, very fast because of the power of the system into therapeutics. And so one of this CRISPR technology has already been proved by FDA, and that is the CRISPR technology that's used in sickle cell disease therapy, that it allows the sickle cell patient to reactivate their fetal hemoglobin, which is the molecule that carries the oxygen around the body, whereas to bypass their defective and adult hemoglobin. So in addition to this FDA-approved technology, there are many more, and then it's currently in clinical trials in different phases of the clinical trial, and look very promising to be in future therapeutics. And thank for the Innovative Genome Institute and University of California, Berkeley, who keep close update on this information. So despite that they are, these technology have to go through all these FDA approval processes, you always hear many of these what's called a CRISPR on demand, personalized medicine. And it is very exciting. For instance, this example just recently reported on this baby named KJ. It was born with unfortunate but ultra rare genetic disease, which made his on this gene called a CPS1 and camoid phosphate synthetase 1, and the mutation renders this enzyme inactive and therefore not able to digest proteins. And so the baby would have a chance, 50% chance of survival, or to have his liver transplanted. And so, about a month period of over a month, that multiple scientists and institutions got together and understood the problem and know exactly what the precise needed genome editing it was needed, and the PBKJ received the base editor that I talked about as a therapy, and is doing very well. Another exciting discoveries are, for instance, Cleveland Clinic has created off-the-shelf CRISPR editing therapy that's targeted to lower the cholesterol and triglycerides. And they use the CRISPR-Cas9 to remove or silencing the proteins that inhibit what we call lipases. And these are the enzymes that's responsible for breaking down these lipid molecules. So reactivating these lipases molecules that's allow the patient to permanently, because you edited the genome, that lower the cholesterol and triglyceride. And so next slides, please. Yeah. So that leads to that I want to introduce in a few slides of what we're researching here in Van Andel, that I studied this unique Cas9 that's currently not yet available. We're trying to fill the gap that there is no CRISPR-Cas9 that can be sensitive to DNA modifications in the genome. So my colleague, Peter Jones and Steve Bailing here at the VAI were the pioneers actually to study the DNA chemical modifications and their importance in biology as well as the hallmark of cancer. So genomes could lose these modifications. They could also gain these chemical modifications. So to developing a CRISPR technology that is able to discern the changes in these DNA modifications in genome, and we argue that they will have enhanced the safety as well as unique applications. So the way the sort of the pipeline of our research is to starting using computing to identify the bacteria which could potentially host such interesting CRISPR-Cas9 enzymes. And once we computationally identify them, and then we could what we called biochemically characterize these genes, these proteins, and characterize their enzymatic activities. And then my group is has this unique advantage. We're using this powerful technology called cryo-electron microscopy, which is another revolution from the older technology, which called X-ray diffraction, allow us to really visualizing these molecules. So in addition to knowing their biochemical properties, but we can also see them. And why is it important to visualize them? That allow us to what we call to engineer them, to modify them, to optimize them to our needs. And I'll show you an example of that. And then obviously, finally, we will use them in the human cells and potentially for therapeutic use. So an example that we've studied over the last few years is a unique CRISPR-Cas9 that's isolated from this bacteria called Acetothermus cellular litocus, or we call it AC-Cas9. We didn't go to the Norris Gare Basin in the Yellowstone National Park, but we were able to to use its DNA and clone them and study them in a laboratory setting. And we discover, yes, interestingly, this Cas9 is able to sensing that the DNA is methylated. And so it'll perform the typical double-stranded break when the DNA has no methylation or chemical modification, but it would prevent the Cas9 from cutting. So the key problem for this unique Cas9 is it is very inefficient. It's much less efficient than the one that's previously isolated, for instance, by Professor Jennifer Doudna, called a SpyCas9. And so we went on to determine its what we call the three-dimensional structures of this enzyme, together with the guide RNA band in black and the two DNA strands that's shown in red and orange. What's powerful about this three-dimensional study, structure study, is we can actually visualize this enzyme in action. And we know that there is this pink parts, and that's one of the what we call domains that's responsible for breaking one DNA strand. And we saw that this domain undergo what we call a hinge motion. So to make the enzyme active, it must move it to the DNA through this wide swinging of motion. And we also determine the structure of this enzyme when the DNA now is methylated. So interestingly, when the DNA is methylated, that's shown on the right, and this pink domain now shown in green, no longer moves. It just stays where it is, and it just doesn't, that's why it doesn't make the double-stranded break. All right. So they gave us the idea that this motion is critical for it to be, activated. So then that allow us to focusing on the site of the motion, or we call the hinge that carries this motion. It's kind of more like the elbow, the motion. So we performed something called protein directly evolution. So to sort of using this analogy of the athletes that has to jump through this high bar, that we create artificial situation, we make the bar higher and higher and higher, then we ask millions of athletes who are different, and to ask them who can jump over. So, and that allow us to be able to identify winners, that we make the bar very high, and then we found the winners. And then these winners has to be the Cas9 that bear some changes in its hinge region. So it makes it stronger and faster. And so you can see what we call the wild type, the native itself doesn't have the activity, while these mutant allow you to perform the function very efficiently. And so we can also test these what we call the catalytically enhanced versions of the enzyme in human cells, that indeed they perform better than the wild type enzyme. So this shows you an example of how important it is to characterize the biochemistry of these CRISPR enzymes and visualizing their three-dimensional structures, so that you can really have a good basis for you to say what part, which section of this enzyme that one can try to improve, and to understand why it is making certain functions that it is not supposed to. So next slides, please. Okay, so how do we harness the power of this methylation sensitive Cas9? For instance, We know the normal cells, many sites are chemically methylated, as this red dot shows, but a corresponding site in cancer cell could have lost it. And we can imagine that we can direct the Cas9 to the site where it lost the methylation in cancer cells, but not in normal cells, and therefore killing the cancer cell. We can also imagine to drive this Cas9 to have the base editing ability to change the nucleotide in a methylation sensitive manner, and therefore, apply the function in the cancer cell specific manner. So we're very excited about this discovery. So the future, how do we continue to harness this technology? We definitely should continue to expand the various CRISPR tools. We definitely should further optimize the efficiency. We really need to evaluate and improve the safety of these enzymes before we introduce into the human cells. One of the critical improvement is to improve how to deliver these enzymes into the human cells at the site of desired site. And so, in conclusion, I hope I have convinced you, CRISPR technology is really, really easy to use, and it is powerful. It is being adapted by nearly all biomedical research labs, and you can talk to any research labs, and there's probably hardly any who don't use this technology. Compared to the other previous genome editing technology, CRISPR is RNA guided or programmable, and it's very powerful, and it can also avoid the immune response. We also anticipating that in the future, the emerging artificial intelligence, it will make it a very important role in further developing in the CRISPR technology. And you may or may not know that these technology in CRISPR-based, which is not characterized as a GMO, has been already used in the changing in the agriculture, in the food that we actually eat. So to conclude, I would like to really acknowledge the scientists who actually did all these work, and a group of scientists that's very inspiring, or ranging from a graduate student to postdoctoral fellows. Some of them are undergraduate students, and I also am grateful for the what we called a cryo-EM core facility here at Van Andel, and as well as at FSU before I moved to here, and some national centers of the microscopy that allow us to perform these study. In addition, Van Andel is just a very supportive, very inspiring place to do research, and all the departments, supporting departments, including all the boards that's listed here, provided really strong support for our research. And I list some of the collaborators that I currently working with. Well, I guess I thank you for your attention and looking forward to some discussions.
All right. Thank you, Dr. Lee, for the wonderful introduction to CRISPR gene editing and your research. Shall we get into some questions? All right, as a reminder to our audience, you may submit questions for Dr. Lee in the chat, which I see we already have a couple in there. So let's get started. You talked about it a little bit, Dr. Lee, and you talked about the different functional modes of CRISPR. And
Yes.
one is mode you mentioned is how you can influence a gene without actually changing the sequence.
Yes.
Can you give a specific example of how you or
Yes.
others are using that mode?
Yes. So for instance, you could attach the CRISPR-Cas9 protein. When I say attach, you have to develop what we call a fusion protein. So you make a CRISPR-Cas9 much longer, so it bears additional protein. And that protein has been, for instance, well studied in human or in other organisms that play a function, for instance, what we call the transcription activation, and that you can bring that molecule to wherever you want, and that to allow that gene to be activated. Or you can, if that part of the protein is what we call a repressor, for instance, and those are well-studied roles of these proteins that they can block the transcription, for instance. That is the step of making from, go from DNA to make RNA. So that way you can, stop making that gene, so you can turn the gene off. So these are the examples of not breaking. Another example are these CRISPR derived technology where scientists have attached a DNA methyltransferase. That's the enzyme that's putting a methyl group onto the DNA, for instance. And then you can guide your this fusion Cas9 as well as this DNA methyltransferase to allow that section of the DNA to be putting on a chemical modification that otherwise lost the methylation, which will influence the, silencing the gene, for instance, if you remove it and you can activate the gene. So these are some of the few examples that you don't even have to make a cut. Simply, Cas9 will act like as a vehicle to deliver these other machineries to the genome of your choice.
Excellent. So you mentioned that CRISPR is being used in a variety of different tools and approaches. You had the two examples from the rare gene, and then also the more over-the-counter example. Once a new treatment is identified using CRISPR or using a CRISPR tool, how fast does it take to get that into patients? For example, if you're going to do something in cancer or the example of that rare genetic disease, right? How long does it take to get to the clinic?
Yeah, I would not say that I'm the expert of the whole clinical trial process, but I was thoroughly impressed with, for instance, the example that I showed that baby KJ was born with genetic disease, where, I think within a month he received the therapy. That's very exciting, but the whole FDA approved process and all that needs to be, go through all the regular clinical trials and etc. But the technology itself is relatively simple to be, put together once you know what site that you wanted to, you wanted to change.
So, what are some common misconceptions about gene editing that you run into when talking with your colleagues or friends and family?
Some of them, I guess, for instance, I think they are questioning about what is CRISPR. They did not know it's actually a bacteria repurposed protein and RNA. So the study of the bacteria led to the repurpose of these molecules that we didn't even knew they exist before. So that might be one misconception. I don't know. Others could be that. I mean, it depends on the person's background,
Mhm.
they don't know what genome it is, then that's much more, there will be much more questions on what the technology is about. Perhaps there are uses of this CRISPR technology used in RNA that maybe people don't know. That you
Right.
don't have to just target DNA. You could, you know, engineer the CRISPR-Cas9 or related CRISPR technology that don't touch the DNA. So not permanently changing the genome, but to, temporarily changing the message itself, the RNA. And
Yeah.
that's another big use of the CRISPR technology too.
Yeah, that's an exciting approach, and it will be
Yes,
interesting to see where we go with that in
that's right.
relates to human health.
Yes.
Mhm. So one of the things that you are trying to do is identify CRISPR tools that read those methylation marks or other marks on our DNA. And you talked a lot about methylation. Can you explain for the audience what methylation specifically
Mhm.
is and how that functions?
Yeah. So in our genome, so these DNA is comprised of these nucleotides. These are the ring-shaped molecules that we have 4 different types on our DNA called adenine, guanine, cytosine, thymine. These nucleotides, normally they are just as these chemical structures. They maintain these chemical structures, but in mostly in eukaryotic cells, such as plants, animals, some of these cytosine, for instance, the nucleotide, can be added an extra chemical group called a methylation. So the addition of a chemical of the methyl group on the C in the genome in multiple positions, multiple locations in the genome, have a profound impact in altering the message that the DNA code. You could making it mostly is for making suppress certain genes or turn on certain genes that's they otherwise the message alone cannot direct that message to. So and then our colleagues' study has found that these methylation can go wrong too. And when they go wrong and when they just lost their normally methylated pattern, or they abnormally gain of a methylation that allow the cell to behave differently. For instance, in cancer, that you have a massive loss of DNA methylation, and then that is a reason to turn on, for instance, some oncogenes and etc.
Mhm.
So it's a very, very important biology that regulate our basically the information highway.
Wonderful. I think that helps explain, right, yeah, the importance of those marks and modifiers on DNA beyond just the A, Gs, Ts, and Cs.
Yes.
So you talked about kind of the next steps, the future, and one of those was AI, right? And these new AI models and for protein folding and structure. How do you see, or how are you leveraging those already to impact the landscape of what CRISPR can do?
That's a great question. So we have already seen some of the AI technology being applied in engineering of the enzymes that they, for instance, you can take the existing CRISPR-Cas9 enzyme, then apply the, AI tools to them and to allow them to be function, stronger, better and safe. Uh, that's much, I guess, computationally much more easily to be realized than we do all these protein directly evolution to come up with a computational model to allow these enzymes to behave better. That's definitely one of a good example of how AI can be used. And I'm sure that AI could also be used to predict and avoid perhaps these off target that that can be that causes the safety concern. Um, because there's a lot of genome data that's there that can be put together, in a language model to learn.
So we don't have a lot of time left. There's a ton of really great questions in the chat that we won't be able to get to, unfortunately.
Mm.
But just to end up, kind of what is it the most exciting to you about your research and kind of the next steps?
Yeah, there's really no ending this research because the bacteria genome, so much bacteria in the world. So in fact, the bacteria outnumber even the bacteria in our human body outnumber our human cells. So there's so much diversity in CRISPR or related technology that can be mined from these microbes that you never know what is the new next CRISPR-like technology. We have seen some of them in early publications now of these new unexpected enzymatic molecules that can perform things we don't yet know yet. So, and another, I guess, excitement is the education. I think it's very satisfying to see, like students all the way from K-12 level, they are excited about the CRISPR technology that we are, in fact, and we're actually making a field trip for our Van Andel Education Institute to allow the middle schoolers or high schoolers to be able to experiment in CRISPR technology. And that shows how simple it is to do and how powerful it can be and how easy it is to understand for these young generation of scientists. And hopefully they can come and make new discoveries. I'm excited about that.
That's really exciting.
Yes.
Well, that is the end of our time. Thank you, Dr. Lee, for sharing your insights into this incredible topic. And thank you to our audience and the VAI community for taking time to learn more about this impactful research. You can visit vai.org to learn more about Dr. Lee's work and more initiatives taking place across the Institute. Make sure to sign up for our mailing list and follow us on social media to stay up to date with the latest news.